Assalamualaikum and good morning everyone. Today we're going to discuss about cellular respiration, okay, but more on the energy carrier, okay, which is for STPM candidates. All right, the ones in your syllabus is the NAD, FAD, H2, and ATP, right? Now, just to recall what you have learned in your secondary education, all right, this is the overall equation for cellular respiration, which is quite the opposite, okay, of that for photosynthesis, right? So we have one molecule of glucose, okay, with 6 molecules of oxygen. That will give you 6 molecules of carbon dioxide with 6 molecules of water and energy. Okay, now, the need of energy in a living organism. Okay, now all living organisms require a constant input of energy. Okay, for body metabolism, for body movement, okay, for cellular active transportation, for cell division process, for production of enzymes as well as hormones, okay, transmission of impulse, control of body temperature. All these needs a constant input of energy. Now, cellular respiration is a process where chemical energy in organic molecule is released. Okay, the usual organic molecule, okay, in living organisms that is being uh, processed in the cellular respiration to release energy is carbohydrate. The released energy from the cellular respiration is transferred to energy carriers like ATP, while some of those energy will lost as heat. Now there are two forms of cellular respiration, okay, which is the aerobic respiration and anaerobic respiration. Aerobic means it has the presence of oxygen, while anaerobic is a type of cellular respiration that occurs with the absence or lack of oxygen. Okay, now let's go into the energy carriers. Now when it comes to this cellular respiration, like the ones that I've talked earlier, okay, the aerobic and anaerobic respiration, that part will be covered in the next video, right? So in this video we focus on the energy carriers. So the first energy carrier is the ATP. I'm sure you are very aware of this ATP. This is an ATP molecule, right, that has the ribose sugar, okay, and then adenosine base and 3 molecules of inorganic phosphate or phosphate, right? Now, adenosine triphosphate is a coenzyme that acts as an energy carrier and a short-term energy storage for cells. Okay. Now, ATP, okay, is derived from nucleotide adenosine monophosphate. This is adenosine monophosphate. Okay, where you have the adenosine pentose sugar, adenosine base with the ribose pentose sugar with 1 molecule of phosphate. This is adenosine monophosphate, or commonly known as AMP. Okay, now an ATP has 3 phosphate group on the other hand, right? 1 ribose and 1 adenosine or adenine base. Now, the energy, right, in the ATP is carried and stored inside the last 2 bonds between the phosphate group. Can you determine where are those 2 bonds? Those last 2 bonds. Okay, it's here. Okay, these are the last 2 bonds where the energy is being carried and kept or stored, right? in an ATP. If we have an ADP, this is an ADP only until here. Okay, in an ADP, the energy is carried and stored only in the last bond between the phosphate group. But in an ATP, on the other hand, the energy is being carried and stored on the last two bonds, okay, between the phosphate group. Boleh ya? Di sini di mana energi itu disimpan dan dibawa. So the energy inside these bonds between the phosphate group is released when these bonds are break. Okay, that's why the breakdown of ATP into ADP will release energy. The breakdown of ATP into AMP will release more energy because there are two bonds between phosphate group that is being broken down. Now, in a hydrolysis reaction, okay, an ATP is hydrolyzed into ADP and pi, or inorganic phosphate, with energy. You can see here how this ATP, right, in hydrolysis reaction, what happens, this ATP, okay, is hydrolyzed into ADP and pi, okay, together with energy being released. Now, one molecule or one mole of ATP is equivalent to 30.5 kilojoule of energy. Okay, phosphorylation is a process where an ADP and an inorganic phosphate, right, are converted into ATP by condensation. Okay, that is phosphorylation. So whenever you see the term phosphorylation, it is a process where ADP and pi become ATP through condensation. Okay, you will come across many times of this word in this last chapter, okay, which is cellular respiration. Now, oxidative phosphorylation, there is an additional word here, oxidative. So oxidative phosphorylation is a process where energy from oxidation of organic molecules during respiration is used to add inorganic phosphate to ADP to form ATP. Okay, and usually oxidative phosphorylation involves redox reaction. Substrate level phosphorylation is a process where inorganic phosphate is donated directly to the ADP to form ATP, or the inorganic phosphate is directly donated to the GDP, and this will form the GTP, right? GDP is guanosine diphosphate, while GTP is guanosine triphosphates. Tri because the addition of one inorganic phosphate to the diphosphate. So the GDP became GTP. So if you're wondering what's the difference between ATP and GTP, right? Now let's take a look at their molecular structure. Very much similar, okay, except that, all right, the base here, the nitrogenous base for ATP is adenosine, while for GTP the nitrogenous base is guanine. Okay, it's guanine. That's why it's guanosine triphosphates. Okay, yang lain very much similar. Ada tiga phosphate molecule di sini. Ini ada pentose sugar. Right. Photophosphorylation is a process where ATP is made using energy from the light. Okay, so now until here, okay, you already learned that ATP, all right, can be formed or is formed by oxidative phosphorylation, substrate level phosphorylation, and photophosphorylation. Right, these three processes here can generate ATP. Now, ATP, excuse me, a bit sleepy. Okay, ATP is used for active transport, all right, where the ATP provides the energy required to move substances against the concentration gradient. And then ATP also used for anabolic processes, where ATP provides energy to build macromolecules from monomers. Example is the linking of amino acids to form proteins. Okay. Now, I did mention about cellular movements earlier, right? So the ATP provides energy for the movement of cilia and flagella. Okay, and then ATP also used for activation of chemicals. Okay, where ATP makes chemicals more reactive. Example, phosphorylation of glucose during glycolysis made the phosphorylated glucose becomes reactive. This part you will learn in detail in the glycolysis. Okay, in the next video, inshallah. Now, just to refresh your chemistry knowledge on the redox reaction. So, in one redox reaction, it consists of oxidation and reduction, right? One uh, one molecule will be oxidized while the other molecule will be reduced. Okay, so oxidation is a process uh, where an atom, right, loses an electron, or that atom receives an oxygen. Okay, so reduction will be the other opposite. So we can generalize, okay, redox reaction like this. Okay, so the atom here receive the loses the electron, so become an X. Okay, so this being oxidized while the other becomes reduced because gains the electron. Okay, so reactants and products. So if these are the reactants, okay, the methane will be oxidized into carbon dioxide, while the oxygen here will be reduced to water. Okay, so we can conclude that the methane, all right, it becomes oxidized, right? So the methane is a reducing agent, while the oxygen is being reduced, so the oxygen is an oxidizing agent. All right, the second energy carrier, NAD plus. Okay, NAD plus, all right. So NAD plus, all right, is nicotinamide adenine dinucleotide, right? Nicotinamide adenine dinucleotide, and this NAD plus is a coenzyme that acts as hydrogen ion and electron acceptor or carrier, and will be reduced to form NADH or NADH with hydrogen ion or NADH with proton during cellular respiration. Okay, so this NAD plus involved in cell oxidation pathways and electron transport chain in the cellular respiration. So we can generally say that NAD plus carries and transfer one hydrogen ion and a pair of electron to the electron transport chain to produce ATP via oxidative phosphorylation. Okay, so you can see how this NAD plus, okay, transfer the hydrogen ion and pair of electron to form NADH. Okay, so if you're wondering why with the excess hydrogen ion here, now this NAD plus, right, when this NAD plus, okay, undergoes dehydrogenase, meaning to say this NAD plus will carry one hydrogen ion, right? Like what I mentioned earlier, as well as a pair of electron. So when this NAD plus carry one hydrogen ion, this NAD plus will become NADH. Sorry, when this NAD plus carry that one hydrogen ion with two electrons or a pair of electron, this NAD plus become NADH. Now there is an excess hydrogen here, excess proton. That excess proton will tag along the NADH. That's why we have an equation here, NADH plus hydrogen ion or plus proton. Okay, NADH, this NADH did not carry this second proton. The second proton is just tagging along the NADH. So one NADH yields 3 ATP. Okay. Now NAD plus is derived from niacin, or commonly known as vitamin B3, which you can get from meat, wholemeal bread, as well as liver, right? Chicken liver, for example, right? Now NAD plus consists of two nucleotides. You can see here the NAD plus here. Okay, consists of two nucleotides. This is the nicotinamide monophosphates, right? This is the nicotinamide monophosphate. Okay, nicotinamide monophosphate, NMP, and this is the adenosine, right? Adenosine monophosphate, AMP. Okay, so apparently these two nucleotides are joined head to head by their phosphate group, right? You can see here, it's here. So you can see how the nicotinamide monophosphate, right, binds with the hydrogen ions, all right, hydrogen ions, as well as the electron. And then you can see how this hydrogen ion here shifted here. So we have an extra hydrogen ion here, right? That's why we have NADH, from NAD plus into NADH because of this, right? So the excess hydrogen ion from here will just tag along the NADH. So NAD plus serves as an electron shuttle during cellular respiration. Okay, electron shuttle because the NAD plus carry and transfer that pair of electrons. So we can conclude here that NAD plus accepts a pair of electrons and a proton from catabolized macromolecules and is reduced to NADH. Okay. Now the third energy carrier, FAD. So FAD is flavin adenosine dinucleotides. Okay, so this is how a FAD look like. Sorry, here, right? Flavin adenine dinucleotides. Okay, sorry, I think, yeah, the whole thing here. Flavin adenine, okay, dinucleotides. So this FAD, all right, is a coenzyme. Okay, now sorry for the confusion earlier. Now this whole thing here is the FAD, all right. This is FAD, sort of like molecular structure. Okay, you can see consists of the flavin, the ribitol, and the ADP. So FAD is a coenzyme that acts as hydrogen ion and electron acceptor or electron carrier, very much similar with the NAD plus. And this FAD, right, when this FAD receives two hydrogen ions or two protons with a pair of electron, this FAD will be reduced to FADH2. So you can see after this FAD receive the two protons with two electrons, okay, this FAD is reduced to FADH2. This is the excess hydrogen or excess proton that attached to the FADH2. It's here, right? Initially none. Okay, so in FADH2, this is where the two proton is. the two protons are, okay, together with the electron. The electron is needed to uh, make the formation of the bond here. So FAD is involved in cell oxidation pathways, which is in the Krebs cycle, which you will learn later on, okay, as well as electron transport chain in cellular respiration. So we can conclude here that FAD carries and transfer two hydrogen ion and a pair of electron to the electron transport chain to produce or to synthesize ATP via oxidative phosphorylation. So one FADH2 will yield 2 ATP. Now, FADH2 is produced in the Krebs cycle, okay, like what I mentioned earlier. Earlier I said that the FAD is involved in the Krebs cycle. So meaning to say FADH2 is produced in the Krebs cycle. And then FAD consists of riboflavin that is bound to the phosphate group of an ADP. Okay. Now, FADH2 transfers its electrons to cytochrome 2, okay, at a lower energy level compared with NADH. So the FADH2 will produce or will yield less ATP compared with NADH. If you recall, one NADH yields 3 ATP, but one FADH2 only yields 2 ATP. Okay, the thing is because the electrons from the FADH2 is transferred to the cytochrome 2, which has low energy level. All right, ATP synthase. Okay, this is not an energy carrier, but it is a part, okay, of a and this ATP synthase is an enzyme. All right, that is a part in the process of synthesis of ATP. Right now, ATP synthase, okay, is an enzyme. Okay, it is an enzyme or a protein complex that is embedded in the inner membrane of the mitochondria. So this is a mitochondria outer membrane, and this is the inner membrane of the mitochondria. And in the inner membrane of the mitochondria is where you can see, all right, or where the presence of the ATP synthase can be found, all right. Now, proton gradient is accumulated when there is high proton concentration in the intermembrane space. You can see here, this is the intermembrane space of the mitochondria where the proton concentration is higher, okay, compared with the matrix of the mitochondria or mitochondrial matrix. Why the proton gradient here is higher is because of the action of the NADH as well as FADH2. This part you will learn later on in the electron transport chain topic. Okay, or from this diagram also you can see how this NADH and the FADH2 carry and transfer, okay, the hydrogen ions, all right, from the mitochondrial matrix into the intermembrane space. You can see how this proton is being pushed out. Okay, it is because of the action of the FADH2 and NADH. So proton will accumulate here in the intermembrane space. You can see here, right? So now there is high proton concentration in the intermembrane space compared with the mitochondrial matrix. So what happens? This proton will diffuse into the mitochondrial matrix from the intermembrane space. Now potential energy is released, right? And that potential energy is used to synthesize ATP from ADP. Whenever this proton diffuse into the mitochondrial matrix through this ATP synthase, what happens? okay, potential energy will be generated by the ATP synthase, and the potential energy will be released by that ATP synthase, okay, to synthesis ATP from ADP with an inorganic phosphates. So until here, okay, candidates or students, you must be able to list out the differences between NADH and FADH2. Okay, so NADH, okay, is the reduced form of NAD plus. All right, that is the nicotinamide adenine dinucleotide. And then NADH is formed during hydrolysis, link reaction, and Krebs cycle. And then NAD plus receives a pair of electron and two hydrogen ions to form NADH with the excess hydrogen ion, right? So class NAD plus receives a pair of electron and two hydrogen ion, but NAD plus only carries and transfer a pair of electron and one hydrogen ion. Okay, let me rephrase. NAD plus terima satu pasang elektron dan dua proton, tetapi NAD plus hanya membawa, okay, carry or transfer one pair of electron dan satu proton. NADH acts as a hydrogen ion carrier and donates the hydrogen ion to the complex 1 in the electron transport chain. And 3 ATP molecules are synthesized via chemiosmosis for every 2 hydrogen ion donated by NADH to the electron transport chain. Okay, for FADH2, there is the reduced form of FAD, flavin adenine dinucleotide, formed in the Krebs cycle. And then FAD accepts 2 hydrogen ions and a pair of electrons to form FADH2. And then FADH2 acts as hydrogen carrier and donates the hydrogen to complex 2 in the electron transport chain. And 2 ATP molecules are synthesized by chemiosmosis for every 2 hydrogen ion donated by FADH2 to the electron transport chain. Okay, so that's it for today. Okay, class. Thank you.